Thank you, everybody. I'll be giving a talk today about a program that I've been coordinating on behalf of the U.S. Geological Survey, which is part of the Department of Interior. Department of Interior is the overall department that has agencies like the National Park Service. So the USGS mission is to provide scientific information to these agencies for decision making. And obviously what we're trying to do here with this new USGS program is to provide information about biosurveillance and specifically aquatic invasive species. So a lot of my talk is going to echo so much of what we talked about in this last session, which is this kind of movement or convergence towards programmatic standardization. Alejandro brought up some great points earlier today. So did Florian. I think the only difference in my talk is I have robots. So that's a good part here. So one of the goals here with this new program I'm coordinating is we're taking the opportunity with an infusion of funds to really try to bring harmonization and coordination to all the different eDNA programs within the USGS. Alejandro had a map of like 30 different centers in Australia that do eDNA. We can have a comparable map in the United States. USGS, last we counted, has over, I think, 15 centers, and it could be as many as 20 plus, because not everyone actually, publishes and lets it know that they're doing eDNA. So we're taking this opportunity with this new program to really focus and kind of clean up our own house, but also to kind of provide a model to the Department of Interior, which has many additional eDNA programs. U.S. Fish and Wildlife Service, National Park Service, Bureau of Reclamation. They all have eDNA programs. We're also using it as an opportunity to provide a template to other federal agencies. And we're also using it as an opportunity to harmonize with Canada. I'll talk here briefly about what we're doing with iTrack. And I do want to point out, I think in the CSA talk, and others have mentioned, we did not invent a new proficiency scheme. We were on the verge of doing it, but then we recognized that Canada was doing it, and so we decided to join up with them. And I think this speaks to the need here that I see with a lot of end users, with that Wild West confusion when they have different agencies, different countries telling them different things. And I see this a lot. I'm based in Montana. I do a lot of work in the Columbia River Basin. I get to interact with a lot of BC and Alberta folks that have great eDNA programs, and we're working with the same managers often. So really the needed opportunity to provide a coordinated message to our end users who are often at the same meetings. And then finally, we like to take these opportunities and travel to this meeting. I have colleagues that are part of this new program in the USGS that were in Australia a week and a half ago, and then colleagues that traveled to Japan. And we're taking these opportunities to really learn how to harmonize with everything else that's happening globally with standards development. So I think this is kind of how we're trying to address the question that was brought up in the last session. What do you do if all these different countries come up with different standards? Well, you become active in the eDNA community and you learn from each other. And you try to pivot and position so you can harmonize your program. So, on with science. So, aquatic invasive species, what is the need here? Well, we know, just like with so many things, if we can detect an invasive early, just like if we can detect cancer early, we have a chance at successful and cost-efficient management before the problem gets too big and begins to spread. So there's this huge need across the world, but right now a huge focus in the U.S. to create early detection rapid response program. We know that it saves money, and in the long term, it will reduce negative effects. But it's hard, and we know eDNA is a great tool. If you think about zebra and quagga mussels, we know plankton toad sampling is useful, but it takes a lot of effort. In lake in Montana, where we had a potential introduction event in 2016, did some work there, and did some work in reservoirs across the West. And we need, tens, if not hundreds, of plankton toad samples to have a high probability of detecting one larval mussel. eDNA, probably tons of samples. So we know it's very sensitive, but that's not enough. Even though eDNA is so sensitive, it becomes quite challenging, whether that's tens of samples, or that's standards development or methods development, to detect these rare introduction events. It really causes kind of that perfect alignment of being at the right time, at the right place, with the right tool. The other problem with eDNA, as we talked about in the last session, is trust. Even if we do have right time, right place, right tool, we still have to convince the end user or the manager that they can trust those results. Meredith alluded to this in his talk, and there's been plenty of papers on the topic over the years. So in addition to kind of providing this great tool set, we need to provide a parallel or complementary tool set that helps end users and managers trust those data. So those are kind of the two big problems that we're trying to tackle here. And recently in 2021, in the fall, there was a big infusion of money that was part of the bipartisan infrastructure law that is going to support a national early detection rapid response program administered by the Department of Interior. It is this multifaceted program that starts with kind of a hotspot analysis. So who do we look for and where do we look? Through a detection phase, which I'll be talking about, through a reporting and decision support phase. So it's a multifaceted framework. The Department of Interior has asked USGS to build out a lot of the tools for this framework, and I've been asked to coordinate one of the tools that focuses on the surveillance component of this larger schematic. The program I'm coordinating is called READI-Net, which stands for the Rapid eDNA Assessment and Deployment Initiative and Network. And like a lot of other programs, iTrack, aquatic, it's this multi-pronged, multi-phase program, because we realize that for these tools to be actionable, we need a lot of steps. There's a long way. There's still a long way to go, and we have to really fit all the pieces together for the data at the end of the day to be actionable. So what we're trying to do with this READI-Net program is a couple of different things. First, we need to make sure we're at that right time, right place with eDNA. So to do that, we're trying to improve the eDNA detection technology. That's where the robots come into play here, because so many places are hard to get to, or maybe the signal is very ephemeral or pulsing, so we need those robots out to be there at the right time. We need to make them cheaper so we can get a lot of them out there and be at the right place. But then we need to figure out how to make those data actionable. How do we make those data trusted? And that's kind of where this talk overlaps with what we've heard previously. Huge focus on the standards and a huge focus on communication and partner engagement. And if we're successful in this program, then hopefully we achieve what we're trying to do with the early detection rapid response, and that is find the invader early, control it, save money, and save costs. So I'm going to walk you through the different components of this program briefly. The first here is that focus on right tool. Over the years, I've been working with the Monterey Bay Aquarium Research Institute, MBARI, to take a lot of the really amazing technology they've been using in the ocean and to deploy it in freshwater systems, specifically to pair it with our stream gauge network at the USGS operates. So trying to combine physical measurements of water with biological measurements from eDNA. What we've learned over the years, fantastic. The more data, the better data. So what we see in this graph here is detection with 0 and 1 on the y-axis. On the x-axis, I chopped out the dates there, but that spans about a 2.5-month period in the summer. We were looking for a rare fish pathogen, or DNA of a rare fish pathogen, and we were using that robotic sampler to collect samples anywhere from every 3 hours to maybe 2 or 3 times a day. And what we do find is that we were able to pick it up a couple of times, and we were missing that with the manual samples where we only went out once a week. So really, the robots really did allow us to be there at the right time. The right time was sometimes early in the morning, sometimes it was at midnight. So that really helps kind of move that right tool at the right time, but it's expensive. I think if you were to buy that robot sampler off the shelf, $125K. And that does not even include the engineers that you need to run it. All I could do was help lift it and get it into the stream gauge. That was really the extent of my expertise. So we're working with MBARI to simplify it, to shrink it, and to make it a heck of a lot less expensive, so we can get it out to the right places across the country. I'll touch upon this briefly here. This is pretty interesting, and what separates my talk from all 100 again is the robots. So what we're trying to do here is to build a very specific eDNA sampler that is focused on filtering and preservation, not in situ analysis. It's going to have 144 sample capacity, two-way communication, so you can fire it off from your iPhone from the office and maybe capture a rain event or some type of sporadic event. It's going to be easy to use and maintain. Hopefully it will be, if it does break, hopefully it won't break. If it does break, a Leatherman and some off-the-shelf parts will be all you need to fix it, versus where it currently is, which is you need Embari or the equivalent of NASA to come in and fix it. And importantly, managers want their information fast. Maybe they don't want to wait until there's 144 samples left. Maybe they want to come in and get it after 10 samples. So they want to maybe visit it on a weekly basis. We're working with Embari to build it in such a way that technicians or whoever else wants to grab those samples can get in and get out in 30 minutes. Currently, with the current robot that we've been using, it's about a 2-day process. So really trying to make this a more usable tool and get it out to many more places. And then the other, I think, redeeming quality here, and again, this goes to the previous talk of standards, it allows us a way to start to standardize field sampling at many sites across the country. So we're really excited about this tool. Should be, we're hoping, in final phase of development in 2025. Next part we're working on here is, well, where do we put these? If we're in a crisis situation and we don't have time to collect pilot data, we all recognize the importance of pilot data to inform where to sample and how intensively to sample. But we don't have time because it's an emergency situation. We need some better models that take advantage of what we know about eDNA taking transport. So we're really digging into creating these biostatistical tools to inform where to get these samplers out in the environment, or where to get humans out that have manual eDNA sampling capability. So really building off of a lot of what's been done in Europe with the Ferrero et al. EDIT models, and trying to come up with a little bit more generalizable frameworks for eDNA taking transport to inform sampling design. Third, and I won't touch upon this because Karen and others have done a great job, digging in on the standards. We recognize that's a critical barrier to trust and implementation. So here we're partnering with iTrack. We are, I think, 7 of the labs that are part of that program, doing the proficiency testing and the intercalibration exercises. So that will hopefully allow us to have these standards that we can use across all these different USGS READI-Net labs. If we have robotic samplers out there that are maybe pumping out thousands of samples, we can now get them to what lab has an opening. So there's not a bottleneck in the lab, because we do ultimately want to be able to turn these samples around as fast as we can. If we have the standards and the proficiency testing framework dialed, then we can farm them out to whatever USGS lab or Department of Interior lab or private lab that has proven proficiency with those standards. And then next we need is a data management system. So right now, every USGS center, every USGS lab has a different way of doing data management. We don't have any way to network with each other. We don't have any way to network with other agencies. So I think someone this morning talked about that data management component and how there's no off-the-shelf way to do this currently. So we are building one. It's going to be based primarily on Esri or ARC products. And the majority of this focus here is actually on QA/QC. So how do we automate as much of the process as possible, from using survey 1, 2, 3 when we're in the field to collect barcodes of the filters, to getting those samples into the lab, to tracking to make sure that there are no errors in the tracking process. How do we get our information from our efficiency testing into here? How do we get the results into here? For the purposes of READI-Net, we will then be sending these data on after they pass all the QA/QC to the USGS non-indigenous aquatic species database, where they would be paired with all the metadata. Finally, this communication component, Alejandro touched upon it. This actually should have been the first one, but it's often easier to talk about at the end. This is all based on partner communication and engagement. So we're working with all of our partners to design each element of this framework, from everything from what do you want in an eDNA robot, how many samples do you want, to what are standards that you will trust, to how you want to view the data, and what are some of the kind of fair and care terms and chain of custody that you want associated with the data. Huge focus here is on that idea of trust and no surprises with the end user, to make everything as transparent as possible from stage 1. So what we hope by 2025 is to have this READI-Net program in implementation phase, and to share it with our partners within the Department of Interior and beyond. but not necessarily to offer it as, we want to offer it as an end-to-end program, but we also want to offer it in kind of this piecemeal approach. So if we do have a partner that already has an existing eDNA program, or already has an existing eDNA lab that they send their samples to, maybe they just want to take advantage of our biostatistical tools. So allow them to kind of grab the pieces that allow them to really strengthen their program versus just to take the whole package as is. So that's the hope, and then our immediate focus here is on aquatic invasive species, but we all recognize eDNA sampling is fairly agnostic. And so in the kind of next versions, we are moving on to metabarcoding and hope to kind of move to teeny species, microbes, human health concerns. But for the near term, it's targeted eDNA approach focused on aquatic invasives. So probably out of time. Just out of time. Yep, and that's all I have. So thank you.